Welcome back, everybody. This is part 3 of the urinary system, and in this lecture we're going to go over the structure and the function of the nephron and the different parts of the nephron, and then specifically the histological appearance of the different parts of the nephron, which can be kind of tricky, so we'll go over the different characteristics of those. This video is sponsored by Doc2Doc, the personal lending platform designed for doctors by doctors. Do you have some big expenses in the near future? Maybe you're moving, applying to residency or fellowship, fixing up your car or home, or starting a new practice. Doc2Doc believes that traditional lenders overestimate the risk of lending money to doctors, residents, and medical students, focusing too much on the challenges of their financial past and giving them insufficient credit for the promise of their financial future. Check out Doc2Doc's personal loan options at doctodoclending.com/DaVinci. All right, so now we're going to talk about the nephron. And so we have a simple diagram here to kind of illustrate the different segments. So here you have the renal corpuscle. This would be the glomerular capillaries or the glomerulus, and then you'd have the surrounding Bowman's capsule, and then you have the lumen of Bowman's capsule or the Bowman's space, and then you move into the first segment here, which is the proximal convoluted tubule, where you have significant reabsorption. About 65% of the reabsorption that occurs in the nephron occurs in the proximal convoluted tubule. Then you make your way down to the proximal straight tubule, which is a conduit to this segment here, these 3 segments here that comprise the loop of Henle. And so you move throughout, and this is a segment has a significant responsibility for concentrating urine. And then you make your way into the terminal segment. So this would be the terminal segment, known as the distal convoluted tubule. So right here, this is the distal border of the nephron, and then you reach the collecting duct, which is kind of this final segment here that actually can receive distal convoluted tubules from multiple nephrons. So multiple nephrons can converge onto one collecting duct, and then this is kind of also responsible for concentrating the urine, and then from here is where it dumps into the minor calyx. So the proximal convoluted tubule, this is the first segment of the nephron extending from Bowman's capsule. Remember, this portion right here is known as the urinary pole. It consists of simple cuboidal epithelium with a microvilli brush border. So kind of to draw out what the cell looks like this. So significant brush border. If you went on electron microscopy, you would see significant microvilli within this brush border. And this is, as you know, to increase the surface area for significant absorption that's occurring here. It's the major site of reabsorption. Again, 65% of the reabsorption of the entire nephron occurs in the proximal convoluted tubule. The way it does that is it carries out transcellular reabsorption of glucose, amino acids, electrolytes, sodium, potassium, I should say small proteins, smaller than albumin, and then water via both active and passive transport proteins. There's a large amount of mitochondria in these cells, in these epithelial cells in the proximal convoluted tubule, because you're going to need ATP for all these transport processes, and as well for obviously the basolateral sodium potassium pump that helps facilitate all these processes. I'll draw a diagram in a second here to help illustrate this. There's also high amounts of aquaporin channels that help carry out transcellular movement of water from the nephron lumen into the peritubular capillaries, because remember, in here you're going to have peritubular capillaries. So if we go to the whiteboard here for a second, we draw out some cells here. So this is our lumen, obviously. This is either side. This is, the epithelial layer. And so on the basolateral portion, which would be here, and then you have the apical, obviously, which is what faces the lumen. And then here, you're going to have an interstitial space here, and then you're going to have the peritubular capillaries. So then here in the basolateral membrane, you're going to have the sodium potassium pump, which is going to move sodium out of the cell, because remember the sodium gradient, the sodium concentration gradient in the extracellular space is much greater than the sodium concentration inside the cell in the intracellular space. And so you create that gradient which drives movement of sodium across the membrane into the cell. On the flip side, you're going to be moving potassium into the cell, because remember, the potassium intracellular concentration is much greater than the potassium extracellular concentration. And so again, the reason you need to use ATP for this is because you're moving against the concentration gradient by moving the sodium out of the cell and potassium into the cell. So you need ATP to power that process. So what happens here on the apical membrane here, facing the lumen here, is let's say you have glucose. Well, to reabsorb glucose, you actually rely on the sodium gradient to drive that. So sodium obviously is going to move passively across this cotransporter. Cotransporter is going to transport multiple different molecules. So you're going to move sodium down its gradient, and then glucose is really just along for the ride. So this is the sodium glucose cotransporter. And so you move glucose down, and then in the basolateral membrane, you just have glucose transporters that can just move glucose from the cell into the paratubular capillaries. Same thing occurs out here for, let's say, amino acids. So same thing, sodium is going to ride down its gradient, amino acids are along for the ride, and then you have amino acid transport in the basolateral membrane, and you're going to move those amino acids from these cuboidal cells into the paratubular capillaries. And so again, that's just to kind of give you an overview of how these reabsorptive processes occur. As far as your histological appearance, like I said, you're going to have significant eosinophilia due to the high concentrations of transport proteins, mitochondria as well, that are all there to facilitate these reabsorptive processes. So if you look at the section here, and this one here, this is a proximal convoluted tubule. In this section here, this is a proximal convoluted tubule. And then here you kind of have a more longitudinal view of one. And you can see that, these are very cuboidal shaped epithelium. It's a single layer, simple cuboidal. They have large central nuclei that you can see here all throughout here. And then what you'll notice is you can see it's kind of ragged on the apical membrane here. And this represents that brush border. So it's going to be really ragged, and that kind of helps distinguish it from other segments of the nephron that we'll go over later. The other thing is it has kind of a characteristic star-shaped lumen. So other sections like the distal convoluted tubule has a very sharp round lumen. This would be the distal convoluted tubule, versus proximal convoluted tubule has more of a kind of star shape where you have like multiple points to it like this. Like that, or say like this. And you can see that here. See how it has multiple different points. It's not necessarily going to be a perfect star shape, but you get the idea, like that there's multiple. It's not a nice sharp round lumen. It's more, it has multiple different points to it. So the other thing is that vitamin D synthesis is a multi-organ process, and the kidney plays an important role. So you have the skin where it starts out, and we mentioned this in the skin lecture. You have UV light that comes in, stimulates the production of vitamin D3. Then it moves to the liver, where it undergoes the first of two hydroxylation reactions. And so in the liver, what you generate is 25 hydroxy vitamin D. I should say vitamin D3. Then it moves to the kidney, where you undergo a second hydroxylation reaction, as we say here, via an enzyme 1-alpha hydroxylase, which is found in the proximal tubule. So 1-alpha hydroxylase. This will yield 1-alpha 25 dihydroxy vitamin D. This is the active form. So you need this final step in the kidney to give you the active form of vitamin D. And what's interesting is parathyroid hormone, in response to low calcium, will stimulate this enzyme. And so it stimulates this enzyme to give you more vitamin D3, because vitamin D, one of the main functions of vitamin D3 is to increase calcium reabsorption in the kidney and calcium absorption in the small intestines as well. So the proximal straight tubule, there's not too much to know about this. We show it here. It connects the proximal convoluted tubule to the loop of Henle. So you move your way down here to the loop of Henle. There's less absorption that occurs here in this region, especially compared to the proximal convoluted tubule. Because of that, it has fewer mitochondria and then a less extensive brush border. Some textbooks, you may actually see this referred to as the thick descending limb of the loop of Henle, but it's not really considered functionally one of the components of the loop of Henle, but just in case you see that in a book or on a website somewhere. So the loop of Henle is a region of the nephron that's really within the medulla, and it consists of three parts: the thin descending limb, which you show here, the thin ascending limb, and then the thick ascending limb here. This is also known as the distal straight tubule, and mainly this is responsible for, it reabsorbs sodium, potassium, and chloride, but this is also more so part of the process of starting to concentrate the urine. The loop of Henle's main responsibility is to contribute to concentrating the urine. So as far as the function for these different segments, the thin descending and ascending limbs are lined with simple squamous epithelium, so much thinner than up here in the proximal tubule. And then reabsorption is much more passive in the thin limbs, so there's less mitochondria present. The thin descending limb is permeable to water, so water can easily move in and out. It's also passively permeable to sodium chloride movement into the lumen. So sodium chloride is not going to move out, but it can move in. Versus the thin ascending limb is water impermeable. So you're not going to be having much water move in between. However, it's passively permeable to sodium chloride reabsorption, meaning you can reabsorb sodium chloride out of the lumen of the nephron. And by removing sodium chloride, that's actually going to help with diluting the urine, because you're going to have less solute, less osmotically active molecules in there. The thick ascending limb carries out secondary active transport for reabsorption of sodium, potassium, and chloride in this portion here. And we'll go to the whiteboard here to show you how that works. So this would be the lumen here. This would be the apical membrane, basolateral. And then here you have a cotransporter known as the sodium chloride potassium cotransporter. And so it's in the name, it's going to move sodium, potassium, and chloride into the cytoplasm of the cell. On the basolateral, you guessed it, you're going to have the sodium potassium pump, moving sodium outside the cell and then moving potassium inside the cell, and then obviously using up ATP to do that. And then on the basolateral membrane here, you're going to have transporters for potassium to exit, chloride to exit, and then the way the sodium exits, you guessed it, via the sodium potassium pump. So essentially this sodium you move in from the lumen gets pumped out by the sodium potassium pump. The other thing I'll point out here is, depending on the location of the renal corpuscle, and which would define what nephron it is, whether it's a coracal nephron or a juxtamedullary nephron, the thin limbs for the loop of Henle have varying lengths. And so for juxtaglomerular nephrons, which are, their corpuscles are going to be found more so at the border here of the cortex and the medulla, they have very long, thin limbs, as you can see here, that begin at the cortical medullary border, which you see here, and then they extend all the way down to the papilla of the medullary pyramid, and then they travel back up to the border here. Versus thin limbs that come from cortical nephrons, so this would be cortical nephrons here. This is juxtaglomerular. So cortical nephrons, they travel much shorter distances. So the renal corpuscle would be somewhere up here in the cortex, and then the thin limb is going to travel down here and maybe just pass over the border between the cortex and the medulla, and then turn back up towards the cortex here. So the histological appearance, sometimes these can be hard to find because they either have cuboidal or squamous cells lining the lumen. They're very thin. Remember, it's in the name, the thin descending or thin ascending limb for the most part, unless you're dealing obviously with the thick ascending limb. But say this segment right here, This is a segment of the loop of Henle. And you can see where the cells, it's a very thin layer. You could say it's cuboidal, maybe even enough for squamous, depending on which section you look at, depending on the cut. You can see here, again, these are much thinner cells here, and you can actually see these kind of look more a little elongated. Maybe this would be kind of more towards the squamous level. And then here as well, you can see the loop of Henle here. They're often found amongst, other collecting ducts and then other tubules as well. And I think what makes them just so characteristic, though, is that the cells are very packed tightly together, and it's a very thin layer of cells, as you can see here, and that's pretty consistent through all three of these. Versus if you remember when we looked at the proximal convoluted tubule, the cell layer was much wider. So the countercurrent multiplier system, as I've referred to previously, is a very complex physiological mechanism, and we're only going to go into very superficial detail because it's something that's more appropriate for a physiology course. But I just want to give you some details so you understand what it is and what its function is. So it's responsible for regulating urine concentration. What happens is that when filtrate leaves the proximal tubule, it's isoosmotic, meaning it's the same osmolarity as the plasma. And so nothing changes as far as solute balance and osmolarity with all of that reabsorption that occurs in the proximal convoluted tubule. So at this point, it's isoosmotic. So if you get asked on an exam what is it? Is it hyperosmotic, isoosmotic, or hypoosmotic? When entering the loop of Henle, this is the answer. So, however, the thin descending tubule, this is permeable to water. So water can leave and enter the interstitium. And then also, sodium chloride can passively be absorbed back into the lumen from the interstitium. So when you remove water, what do you do to the concentration? Well, you further concentrate it. You make it hypertonic because you're removing water and increasing the concentration of those solutes. When you add solute in, such as sodium chloride, you also concentrate it because you're adding more solute in. So at this point, you've created a hypertonic filtrate. And so at this point, at the tip of the loop of Henle, you've yielded a hypertonic fluid. Now, as you start to go back up in the thin and thick ascending tubules, remember, these are impermeable to water. So the amount of water is going to stay the same. However, sodium and chloride and potassium, especially here in the thick ascending limb, remember with that sodium, potassium, chloride co-transporter, is going to reabsorb these. So water stays the same, and then you're removing solute. So if you're removing solute, then at this point, and remember this is a very significant amount of sodium reabsorption. Remember, it's 25% of all of the sodium reabsorption in the nephron occurs here in the thick ascending limb. So because you could maybe wondering, well, how do you go from a hypertonic, and then as I say here, when filtrate leaves the loop of Henle, it's actually hypoosmotic. And so when you get up to this point, you've removed so much sodium, and remember, you have left the water level the same, that at this point, when leaving the loop of Henle, it's hypoosmotic. So that can be a test question as well, is what's the osmolarity of the filtrate when leaving the loop of Henle. Now, what's important to know is that antidiuretic hormone, or ADH, can stimulate the distal tubules in the collecting duct to become very permeable to water entering the interstitium. So although at this point it's hypoosmotic, meaning it's less concentrated, if you have ADH that comes in and stimulates these distal tubules, you can actually stimulate removal of water if you need to further concentrate the urine. And so if you need to concentrate the urine, that would be, ADH is a hormone. It's released from the posterior pituitary, particularly in situations when you're volume down. And so if you want to preserve fluid and preserve volume and don't want to excrete as much fluid, you can reabsorb it here in the kidney. And that's, again, another example of how the kidney contributes to fluid balance. Lastly here, we've mentioned these before, the vasorecta capillaries. So these are, those thin capillaries that are coming down here as well. These are responsible for essentially removing all this water and solute that are reabsorbed into the interstitium. So the distal straight tubule, we've talked about this a little bit before, but just to give you a few more details, it's also the thick ascending limb. So this is also this portion right here. Remember, this is where that sodium potassium chloride co-transporter is. Again, as we say here, it begins at the border between the outer and inner regions of the medulla. So this is down in the medulla still. Consists of simple cuboidal epithelium. These cells, they're smaller and less eosinophilic compared to the proximal tubule. Although a significant portion of sodium reabsorption, not as much cellular transport involved as the proximal tubule. And then the nuclei are found actually in the apical region of the cytoplasm. This is kind of a unique feature. There's also a portion of this that travels through the renal corpuscle between the afferent and the efferent arterioles. So if you have the arterioles on either side, and this is a specialized region of the nephron called the macula densa, and this is responsible for regulating sodium balance, and it's part of the juxtaglomerular apparatus, which we'll talk about at the end of the lecture, because you kind of need the whole picture of the nephron to fully understand its function and how it works. So we'll get to that later in the lecture, so hang on. Then the distal convoluted tubule. So after you get through the distal straight tubule, then this is the terminal end of the nephron. Remember, the nephron ends here. So this is a short segment and extends, like we said, from the distal straight tubule to the collecting duct. It's responsible for reabsorption of sodium and bicarbonate. So it'll reabsorb sodium and bicarbonate. And then it'll actually secrete potassium and ammonium. Now, what's important is there's a hormone called aldosterone, which is secreted from the adrenal cortex, and this causes an increased reabsorption of sodium and an increased secretion of potassium. So it's going to increase this and increase this. It's typically released in situations of low circulating volume. We'll talk about that in a few slides here when we talk about the renin-angiotensin-aldosterone axis. Bottom line is it's going to reabsorb sodium. Remember, where sodium goes, water follows, so it's also stimulating reabsorption of water, and the whole goal here is to increase effective circulating volume and raise blood pressure. As far as the histological appearance of the distal convoluted tubule, the best way to do this is really compare it to the proximal convoluted tubule, which often in these sections you'll see them right nearby. So it's going to lack a brush border, and so if we actually look at the section here, this is the distal convoluted tubule, and right here, nicely for us, is a proximal convoluted tubule. So if you look, it's much more smoother edges versus this kind of ragged edge here. Relatively smaller in size. The cells are smaller, as you can see here, compared to the ones in the proximal convoluted tubule. They have more pale cytoplasm, and you can appreciate that here. If you compare here, there's much more proteins involved, much more mitochondria here. It has more of a round lumen versus this star-shaped lumen that you see in the proximal convoluted tubule. And then you also appreciate this here where you have closely spaced nuclei versus the proximal convoluted tubule. And so if we go through some other sections here, this is a distal convoluted tubule here, same thing, it's much more pale, it's much more round, closely spaced nuclei. Again, if we show you a section here, another good example of it here, those nice smooth edges there. So the collecting tubules or collecting duct, they begin at the distal border of the distal convoluted tubule. So this is where the nephron would end, and then you begin forming the collecting tubules and collecting duct. And then, as we say here, multiple nephrons, as we show here, via their collecting tubules, drain into one cortical collecting duct. Collecting tubules, then they drain into the collecting duct. And this occurs in the medullary rays. This is where the collecting duct begins, in the cortex, and then as it continues down into the medulla, it becomes the medullary collecting ducts. So it's just named for where it is in the region of the kidney, whether it's in the cortex or the medulla. Either way, the epithelium in the medullary collecting ducts transitions to more columnar. So up here, it's going to be more cuboidal. And then let's say, here it becomes the medulla. Here it's going to, it's more cuboidal. Up here, then it's going to transition to more columnar. Really, the cells, you can think of it, they get taller and taller and taller as you continue to go down. Then you have the medullary collecting ducts, also known as the papillary ducts of Bellini, that drain the filtrate at the curbosa into the minor calyx. And so that's again at the papilla of these medullary pyramids like this, and then you have the minor calyx here. There's two types of cells in the collecting duct. There's principal cells, also known as light cells, and then intercalated cells, also known as dark cells. The light and dark comes from how they appear on microscopy. So first, the principal cells, these are the more numerous cell type in the collecting duct. These are represented in blue here. And then the green here are intercalated cells. So these cells, they function to reabsorb sodium and chloride and then water as well. So they can pull water out, help concentrate the urine. They can also secrete potassium. They have a light cytoplasmic appearance, hence their name, also known as light cells. They have a primary cilium present, as you can see here in the diagram, and this is on the apical membrane to mechanically sense the flow of filtrate through here. Also, antidiuretic hormone. This is the main action spot for antidiuretic hormone. It can significantly increase the water permeability of these cells by adding a high concentration of aquaporins. It essentially stimulates the addition of aquaporins to the apical membrane, which then allows for a significant amount of reabsorption of water. This is to help concentrate the urine. This is also antidiuretic hormone, as we mentioned, is released from the posterior pituitary gland, and it's released in response to low circulating volume. Antidiuretic hormone is also released in response to hyperosmolarity of the plasma. So to help normalize the osmolarity of the plasma, it'll increase reabsorption of water to help normalize that. And so that's another function as well, in addition to just trying to reestablish volume. The intercalated cells, these are found between groups of principal cells. So you can see that here in green. They have high numbers of mitochondria, which results in their darker appearance, hence their name as dark cells. They have a more extensive apical microvilli, so they have a little more folds like this. They have more microvilli like this, and they function to modulate the urine pH, so they're really important for acid-base balance. So they'll secrete acid or bicarbonate depending on how you want to modulate the pH of the urine. Again, like the countercurrent multiplier, that's regulating the acid-base balance of urine and of the entire body is an extremely complex physiological phenomenon, and we'll leave that to more of a physiology course. So as far as the histological appearance, they have a low columnar or columnar appearance. You can see that here. This is a great section of it right here. So we'll circle here for you. And you can see how they're a little bit taller here. They have a very light cytoplasm, and you can see that. Really appreciate that here if you compare to surrounding tubules here. And they have also distinct cell boundaries. You can see that here. Notice these lines like this. And so that's another kind of characteristic you can see here again, and that can help you identify these on a section. So this again right here is a collecting duct as well. All right, so we're going to close out this lecture by talking about some of the endocrine regulation of the nephron, especially in response to decreased blood volume, decreased blood pressure, and kind of responses that affect the nephron. So this all starts with a region known as the juxtaglomerular apparatus, and it's composed of the macula densa, which is a portion of the distal convoluted tubule, the extraglomerular mesangial cells, and the juxtaglomerular cells of the afferent arteriole. So if we go down here, here's a renal corpuscle. Again, we're going to have our afferent arteriole, and then we have our efferent arteriole, and then we have our extraglomerular mesangial cells, which are essentially that connective tissue and supportive cells found between the afferent and efferent arteriole. Then you have the juxtaglomerular cells, mainly found in the afferent arteriole. There's some found in the efferent, but they're much more higher concentration in the afferent. And then you have the macula densa, which is a region of the distal convoluted tubule, which is found actually kind of in this region here between the afferent and efferent arteriole. So this is our macula densa. So first, the juxtaglomerular cells. These are specialized vascular smooth muscle cells that synthesize and store an enzyme known as renin in vesicles within the cell. So if we come down here, again, it's going to be mainly found in the afferent arteriole. And you have the juxtaglomerular cells. And what they do is they release renin in response to arterial baroreceptors found in the afferent arteriole that sense decreased circulating volume or decreased blood pressure. So baroreceptors, they sense pressure. So if they sense decreased distension of the vessel, which would be a result of decreased fluid flowing through it or decreased pressure, then what's going to happen is that's going to stimulate renin secretion. So decreased blood pressure or decreased circulating volume. Stimulate these cells to then secrete renin. And that's the first step in the renin-angiotensin-aldosterone axis, which we'll talk about in more detail in just a couple slides. Remember also, just to refresh you, you have sympathetic nerve stimulation that can result in constriction of this afferent arteriole, which then would decrease blood flow through the glomerular arteriole, the glomerular capillaries, thus reducing filtration. The macula densa, which again is a portion of the distal convoluted tubule. It's kind of a region with specialized epithelium. It's found again about right here between the afferent and efferent arteriole. And what it does is it senses sodium concentration and also volume of the filtrate within the nephron. And so the reason why it's found in this particular area, as you'll see in a second, is because in response to, say, decreased sodium concentration within the nephron, the macula densa stimulates first vasodilation of the afferent arteriole. So through secretion of molecules that will help stimulate vasodilation of the afferent arteriole, what that helps to do is increase glomerular capillary hydrostatic pressure as a result of increased blood flow. So then you're going to increase filtration through, and that's, hopefully increasing filtration of sodium through there as well to increase the sodium concentration in the filtrate. The other thing it's going to do is you're going to have these juxtaglomerular cells. It's going to stimulate release of renin. So those are the two main effects. In a way, it's kind of an indirect way to measure the plasma sodium concentration, because if you have decreased plasma sodium concentration, you're not going to be filtering as much sodium through, and thus that's going to be sensed by the macula densa. So now the renin-angiotensin-aldosterone axis, the best way to explain this is by its response to decreased blood volume or decreased blood pressure. So if you have decreased effective circulating volume, you could also include decreased blood pressure for this. What you're going to do is stimulate those juxtaglomerular cells in the afferent arteriole in response to decreased distension of the vessel sensed by those baroreceptors to release renin. Renin is going to stimulate the cleavage of angiotensinogen, which is a protein synthesized in the liver, to angiotensin 1. Then angiotensin 1 is going to get converted into angiotensin 2 by an enzyme called angiotensin converting enzyme, or ACE, which is found in very high concentrations in the lung vascular endothelium. Angiotensin 2 has two primary effects. One, it's going to cause increased vasoconstriction, which is going to cause increased peripheral vascular resistance. Which results in increased blood pressure. The other thing it's going to do is increase aldosterone secretion from the adrenal gland, which then, as we talked about, increases sodium reabsorption and potassium secretion, mainly in the distal convoluted tubule. What that's going to result in is then increased sodium reabsorption, but then also increased water reabsorption, because remember, wherever sodium goes, water follows, which will then increase blood volume, and then in turn, hopefully increase blood pressure. The other thing is that ACE also stimulates the breakdown of bradykinin. Bradykinin is kind of like histamine. It's an inflammatory stimulant, so it stimulates edema and other inflammatory responses. So that's just kind of a side fact to know. Now, in response to increased blood volume or increased blood pressure, that's going to obviously distend those baroreceptors distend the vessel, which will be sensed by those baroreceptors. And so as a result of that, you're going to have decreased renin because you're going to inhibit this. If you have decreased renin, you're going to have decreased concentration of angiotensin 1, decreased concentration of angiotensin 2. As a result, you'd also have increased bradykinin. That's not as important to know, but just keep that in mind. Decreased vasoconstriction or even vasodilation. And then also decreased release of aldosterone, decreased sodium reabsorption, decreased potassium secretion. The last thing we'll talk about here is antidiuretic hormone. We've mentioned this several times throughout the lecture, but just to kind of tie all this stuff together, it's released by the posterior pituitary in response to plasma hyperosmolality. That's the main driver. It's not decreased blood volume. The only exception to that is really hemorrhage. If you have like significant loss of blood volume, you can have stimulus of antidiuretic hormone. It's more when you think of antidiuretic hormone, you want to think of balancing out the osmolality of the blood. So if you have hyperosmolality of the plasma, what you need to do is increase your absorption of water or reabsorption of water in the kidney so that you can balance out that osmolality. So as a result of that, as we talked about, it stimulates aquaporin insertion in a number of different areas. So in the apical membrane of the ascending loop of Henle, so it can do it here, distal convoluted tubule, it can do it here, and then where the main one is that you really want to think about is in the collecting duct. This is the primary site of action. This is where it's going to have the most significant effect. And so you're going to reabsorb a lot of water. What that does is that increases the concentration of urine, because you're removing fluid, so you're increasing the concentration of solute. That's also decreasing urine output. Thank you so much for watching this video. Please like the video and subscribe to our channel to check out more histology videos and other medical education videos.